morning, everybody. Thanks for inviting me here to share some of the work that we've been doing. Just to kind of step back, my life exists actually because my half-sister's mother passed away from metastatic breast cancer, which is kind of wild to think about. And just the time that was occurring, this was the days of pre-mammography screening. And I feel like my life has set me up to be able to talk to you all. I'm sorry, I'm a little emotional. I just haven't slept so well. But anyways. Oh, thank you. But I say that because I also feel like a success story because of all the advances that have happened since I've been born in '87. So anyways, and also in that layer, my biological mother also had breast cancer, but did not have any genetic mutations. And I think it was always my life's goal to figure out why. And, so that's where I am today. Thank you so much. To tell you all a little about this journey, why I think this is so important and relevant to those here today. So, okay, just a quick overview of this talk. I'll go really briefly about the breast cancer burden. I'm an epidemiologist by training, so I present a lot of statistics. So if there's something that I present to you that you need clarification, please let me know. After I talk about that, I'll go into some of the history behind some of the risk factors that we know are well established for breast cancer, but yet the major gaps that we have in establishing risk factors for breast cancer subtypes. And then I'll focus on some of the work that I've been involved in on radon and environmental exposures, also in the context of Kentucky. Oh, I have nothing to disclose. I wish people would fund me. Here's some the most recent statistics from the American Cancer Society about the large burden of breast cancer in the United States. As you all probably are already well aware, breast cancer is the most commonly diagnosed cancer in females. Each year, it's estimated that there are over 300,000 women diagnosed with breast cancer each year. And that's presented up at the top in blue outline. And in the bottom, we have the estimated deaths per year for female breast cancer, where each year it's estimated that around 42,000 females die from breast cancer. So obviously, in this crowd, we know how burdensome this disease is, both in terms of statistics and all the lives that those individuals are impacting. This is where, I love to talk about breast cancer as a success story because, what my half-sister's biological mother never got to know was her breast cancer subtype that may have contributed to her survival. Whereas when my mother got her diagnosis, she had a much more favorable tumor. So, we classify breast tumor subtypes based off of hormonal receptors like estrogen receptor and progesterone receptor. And then we also have our HER2, human epidermal growth factor 2. And the 5-year relative survival rates are very different based off of the tumor diagnosis. So it's quite good prognosis for women who have hormone receptor positive tumors. Around 92% to 96% will survive 5 years. Whereas for the hormone receptor negative tumors, the survival is worse with triple negative cancers. That's the hormone receptor negative, HER2 negative, having around 78% 5-year survival. What I also want to point out in those bar graphs is luckily the more commonly diagnosed cancer are those more favorable tumors, those hormone receptor positive, but there are great racial and ethnic disparities in the types of cancers females are being diagnosed with. And you can see sorry, I don't have a laser. Actually, maybe I do have a laser pointer. Okay, I'm not gonna do that. Maybe I can do my arrow. Okay, so in this bar, okay, you can't see my arrow. And in the second column, that's the bar graph for Black females with breast cancer. And you can see in the dark gray, that is the hormone receptor triple negative breast cancer. So 19% of breast cancers from Black women are those most aggressive tumors. Whereas in other racial groups, it's different. So why are they different? It could be explained by differences in risk factors. But before I jump into that, I want to just define a risk factor. So the National Cancer Institute's definition for a risk factor is something that increases the chance of developing a disease. Some examples of risk factors for cancer are age, a family history of certain cancers, use of tobacco products, being exposed to radiation or certain chemicals, infections, and certain genetic changes. And we've actually known a lot about some of these reproductive risk factors since the early 1900s. This is Dr. Janet Lane Clapin from England, who conducted the first case control study, which is a major epidemiologic study design. And it was actually on reproductive risk factors for breast cancer in England. And while she wasn't using the term risk factor, she was using the term antecedent condition, so something that comes before the disease. And her goal was to compare antecedent conditions from a sufficient and suitable series of cases of cancer of the breast with controls defined as women with conditions of life that were broadly comparable to those of the cancer cases, but who had no sign of cancer. And this was across multiple centers and hospitals in the UK. And what she observed were some of what we now know as established risk factors that women who have children have a lower risk of developing breast cancer than women who don't have children. And in this time, age at marriage was synonymous with age at first birth. Where she also observed that an older age at first birth or marriage was positively associated with having higher risk of breast cancer. So we've really known about this for a long time, and in the 1970s and 60s and 70s, other interesting epidemiologic data came out about related to migration studies. So here on the x-axis you have age, and on the y-axis you have breast cancer incidence, and obviously as age increases, so does breast cancer for all these groups. The black line at the bottom represents breast cancer incidence in Japan in 1966. They had registry data in Japan in that particular area at that time. The top dashed red line are white females in San Francisco in 1969. And what they observed in these migration studies is that females who had immigrated from Japan to San Francisco and to Hawaii adopted the breast cancer incidence rates of those of their counterparts. And recall that this is prior to screening. So this is suggesting that it's not about a change in genetics in this composition, it's something about a change in the environment that led to this upward shift towards that of the host country. So that's the historical interlude, but if our established breast cancer risk factors are things like demographics, like older age, non-Hispanic white race, and non-Hispanic black race and ethnicity. We know that family histories of breast cancer is a strong risk factor for breast cancer, as well as certain genetics, especially BRCA1 and BRCA2, as well as high mammographic density, and having a history of benign breast disease. Now, some of those you can't really change your age, your race, your history, or your genetics yet, or your mammographic density, but some of the more modifiable risk factors that one can argue how modifiable some of these reproductive risk factors actually are, but some of those are being nulliparous as a risk factor for breast cancer, having an older age at first birth, not having breastfed, having an earlier age at menarche, and an older age at menopause. Often these are really tied to estrogen mechanisms, as well as some of these lifestyle factors like having a higher postmenopausal BMI. Actually, there's a paradox with breast cancer risk where lower early life BMI is a risk factor for breast cancer. The use of hormonal therapy and consuming more alcohol and having low physical activity. Also, we know that diethylstilbestrol exposure, which is no longer really used anymore, but this was used to help women during pregnancy with, pregnancy symptoms. But that became a cause of vaginal and breast cancer, so it's no longer really prescribed. The last one that I'll mention is having extremely high doses of ionizing radiation exposure that I'll come back to a little bit later in the talk, but especially if you're exposed early in life to these high doses of ionizing radiation. So what we've established these breast cancer risk factors. You can get your Gale model or your Tyrer-Cuzick model, and a lot of these risk factors are in those models to predict risk of breast cancer. But these are really mainly reflecting risk factors for luminal A or hormone receptor positive, HER2 negative tumors. Because they are the predominant tumor and that is prevalent in the or incident in the population. So colleagues Molly Bernard, Caroline Bucky, and Rula Tamimi published a really great a systematic review of all the different risk factors for breast cancer subtypes specifically. And I'm pointing out here in green that breastfeeding is the one that is a preventative factor for luminal A, luminal B, as well as triple negative. If you also look at the column under luminal A, you can see there's a lot more, the more positives, the more minuses, it means the strength of the association. So we know a lot more about risk factors for luminal A breast cancer. For triple negative breast cancer, this includes reproductive factors, and family history, alcohol use, some of the modifiable ones. But really, breastfeeding is the only preventative risk factor, strong preventative risk factor for triple negative breast cancer. So, given that we know a lot about these well-established modifiable breast cancer risk factors, we know that we know a lot about hormone receptor positive breast cancer risk factors. We know much less about risk factors for estrogen receptor negative disease. But given that there are very few known modifiable risk factors for estrogen receptor negative disease, and given its relatively poor prognosis due to more limited targeted treatments, identifying novel and risk factors for ER-negative breast cancer is vital to prevent the disease, particularly among women who are disproportionately burdened by estrogen receptor negative disease. And we also need to think about non-hormonal risk factors as well, which have largely not been explored to the extent that so many of these hormonal risk factors have. So I want to go back to that notion of the high-dose ionizing radiation, finding that we know is an established risk factor for breast cancer, and it stems back to there's a study in 1994 by Tokunaga and colleagues looking at the cohort of atomic bomb survivors, so people who had survived the atomic bomb. It's called the lifespan study. And on the x-axis, we have the age at which they were exposed to the atomic bomb. And then on the y-axis, we have their excess risk of breast cancer. We don't know what subtypes in this study, again, because it was 1994. But women who are exposed in early life, again, sorry I don't have a pointer, but exposed at ages 0 all the way through maybe 35, had the highest, they were exposed to the atomic bomb and they had the highest risk of developing breast cancer. I did that, that's nice. Another study was conducted was published in 1989, but this took place in the 1930s, 40s, and 50s for women who were being repeatedly exposed to fluoroscopy treatment or radiation to the chest to treat tuberculosis. And they found that women who were, girls who were exposed to that repeated fluoroscopy treatment for tuberculosis had, 4.5 to 2 times higher risk of developing breast cancer later in life. So this was a particularly relevant time window of exposure, being exposed to high-dose radiation. I use this as a segue to now talk about some of the research that I've been involved with, which is radon, which is also a form of ionizing radiation, but at much, much lower doses. And hopefully we never have to experience an atomic bomb or these disasters of high-dose radiation. But radon enters the home mainly through cracks in the foundation in a basement, but can also be found in well water and in water. So I want to talk a little bit more about that. So again, radon is a naturally occurring form of ionizing radiation. It's found in geologic rocks. It's a decay of uranium. And radon, for the general population, is the major source of radiation exposure. So here we have a double-stranded helix of DNA, And when radon is decaying, it releases what are called alpha and beta particles, and that's these really strong particles that can break, cause double-stranded DNA breaks. So here is a picture of that double-stranded DNA breaks, and if you get enough of these, it can cause cancer. Here on the right, we have a figure of just showing what the decay of radon, going from radon to polonium to lead, and I'm not going to do my periodic table of elements here, but you can see that, that this is the decay, and it's releasing these, radon is releasing these alpha particles. So why might radon be relevant to breast cancer? Well, we know high-dose ionizing radiation is a known breast carcinogen. Other studies have published that low-dose ionizing radiation in human breast cell lines induce chromosomal instability, increased cellular proliferation and invasiveness, and altered expression of DNA repair genes. What's also really important that I don't think people really know about radon is it's fat soluble, so it tends to accumulate in the more fatty organs, which is the breast. So here's from Kendall and Smith, published in 2002, showing that there is actually radon detected in breast tissue. So here in this column on the far right, that's the radon isotope. Here you see levels of 1.2 in the lung, and the next highest organ with the highest dose of that radon isotope is the breast. Interestingly, there have actually only been a couple animal studies, so beagles that were exposed to radium, which is a parent of radon, had greater risk of developing malignant mammary tumors, and then rats who were exposed to estrogen and ionizing radiation had hyperproliferation of those mammary glands and induced a pathway that's called P38 mitogen activated protein kinase. I put that in bold because this comes back a little bit later. So this is kind of why we might study this, and my colleagues and I set to study this within the Nurses Health Study cohorts. So these are fantastic participants that have contributed so much to the health of women in the United States. But the Nurses Health Study started in 1976 of 121,700 females who were ages 30 to 55 at that time. And every 2 years, since they were enrolled in 76, they were given questionnaires asking about new diagnoses, risk factors, medical history, reproductive lifestyle, and health behaviors. For the study, they continued to follow these women through 2018, but for the study that I'm presenting to you, we followed through 2014. We also, this map up here at the top is showing the geographic spread of the Nurses Health Study participants. So it started in, I think it was 7 states, I believe, but now they reside all across the contiguous US. The Nurses Health Study is very similar, but it started in 1989 with 116,000 females ages 25 to 42. Same idea, every 2 years they were getting updated. So because they were getting mailed a questionnaire, we know their address and can geolocate those, assign a latitude and longitude. That allows us to study the environments in which these participants live. So my colleague Trang Vo Pham, she worked with developing county-level radon estimates, which is what this map is that you see here, where counties in dark red have the highest levels of radon, whereas counties in the yellow have the lower levels of radon. And this was based off of measuring radon in the home for 115,000 measurements, and they also utilized the EPA's Environmental Protection Agency's state and national residential radon surveys, and did a model to incorporate things that we know predict radon, like soil concentrations, geology, housing characteristics, and more. And what we did is we linked those participant addresses over top those maps using GIS, Geographic Information Systems, so that we could estimate all these participants' county-level radon. And since we had followed them up for the development of breast cancer, we knew that's the research question that we're asking. Do women who live in counties with higher radon have higher rates of developing breast cancer subtypes? So here's our results. This is all published. It's actually kind of old now, but on the figure on the left, we have adjusted hazard ratios, and there's a dashed line across 1. So if those squares are above 1, that means people had higher rates of developing breast cancer than the lowest radon group. So here in green, we are looking at rates of developing estrogen receptor negative disease. So we see here that it's elevated for those that live in the counties with the highest radon levels compared to the women who live in the counties with the lowest radon level. And this was accounting for known differences like age, race, and many other things like SES. I have a couple notes I can tell you about that. But what I also found really interesting is as the breast cancer subtype became better and better classified to triple negative, we observed that women who lived in the counties with the highest levels of radon had 1.52 times higher rates of developing triple negative breast cancer compared to the women who lived in the lowest radon counties. Also, interestingly, we did not observe an association between county level radon and estrogen receptor positive disease. Of course, this was county level. We don't actually know if the participant had high radon in their home themselves, and that's where we are at today to try to, I'll tell you a little bit more about that. We also further evaluated the breast tumor tissue of these participants. So I forgot to put up this, but we had formalin-fixed embedded breast tissue specimens, and we ran a gene expression profile of 17,000 genes and looked at pathways that were related to radon exposure. And what we saw is that women who had lived in the counties with the highest radon had upregulation of, again, that P38 MAPK signaling pathway, and this was specific for the estrogen receptor negative tumors. So I think hopefully the next decade of my life I can tell you more about this story because I think there's a lot to explore here. So then I came to Kentucky and was learning about breast cancer burden in Kentucky. Here is data from the Kentucky Cancer Registry where you can look at rates of many different cancers across Kentucky counties. The map on the left is counties with high breast cancer incidence, just all types of subtypes. And then when we look, and this was from 1995 through 2018, but when we look at triple negative breast cancer, that really didn't get rolled out across the state until 2009. So I'm showing you county level rates of triple negative from 2009 to 2018. But the big idea here is there's a lot of spatial variability in breast cancer incidence across our counties. And the goal for the next stage of the career research endeavors is to look at radon levels. So this is a map of Jefferson County's radon distribution. I forgot to mention that the EPA recommends you get your home remediated if your radon level is above 4 picocuries per liter. If you were to ask the WHO, that threshold would be 2.0 picocuries per liter. We worked with Louisville Metro Department of Public Health and Wellness. They routinely used to actually be free for every Kentuckian to get their radon tested in their home. It is no longer part of the Public Health Cabinet of Health and Family Services, so I'm not sure if it's free anymore. But when we did that, we observed that 33% of the homes being tested in Louisville were above that EPA threshold. And that also corresponds to the highest quintile for those county level results. But what I want to say is currently there are no studies of personal exposure to radon. We don't have a study yet of participants' radon in their home and whether or not that impacts their breast cancer development or not. We are working now to attempt to initiate the Louisville Environmental Assessments pilot study in breast cancer. We're in the process of IRB approval. Remind me to update you a little bit later. Okay. Here's a separate set of research that we conducted. This was published last summer about identifying breast cancer hotspots in Kentucky and their environmental correlates. With the Kentucky Cancer Registry data, we there were 77,679 breast cancer cases diagnosed in Kentuckians from 1995 through 2018. And the Kentucky Cancer Registry, of course, with IRB approval and the appropriate protections, will give you de-identified data like demographics, tobacco use, family history, marital status, and health insurance, and information on tumor receptors, vital status, diagnosis states, treatment, and if you apply for a level 3 protection, you are given a geocoded address of the participant when they were diagnosed. And that allows us to conduct these linkages to environmental databases. So what we first did was want to look at rates across Kentucky at a smaller geographic unit than county, because I don't know if how many people live in Jefferson County, but you might imagine that breast cancer rates change across Jefferson County that we don't see in those county level maps. So the purpose of this work was to look at the census tracts. That's a much smaller geographic unit, particularly in urban areas, that includes about 1,000 people. So we calculated age standardized breast cancer rates in Kentucky's census tracts and used a fancy spatial statistic called the Geddes or GI star to identify areas of high breast cancer rates and not due to differences in age across census tracts. After that, we then described those the people who lived in these areas with high breast cancer rates in terms of ambient heavy metal pollutants, as well as demographic, social, economic, housing, and lifestyle characteristics. So here is our breast cancer hotspot map that was published last year, where you can see Kentucky's 1,115 census tracts. And so we get a much smaller geography. Those census tracts that are in black, that's where nobody lives because it's either, or the population size is too small to calculate a rate because it's either an army base or an airport. So you can see up in the top left Louisville, so that black census tract is the airport, and the other census tracts that are in red were these statistically significant spatial hotspots of cancer. I think we can use these maps to help us potentially target populations that might benefit from screening programs at a smaller geographic resolution than, say, counties. And so, this is Louisville, Lexington, and southeastern Kentucky around McCreary County, I believe. So we looked at characteristics of the people who live in these hotspot census tracts, and the females who lived in those breast cancer hotspots were slightly older, more likely to be Black, single, ever smokers, and use public health insurance like Medicare or Medicaid. And also the cases who lived in the hotspot resided in neighborhoods that were more urban and had more higher levels of poverty. With the ambient data, I apologize for forgetting to include a map of ambient cadmium and arsenic and nickel throughout Kentucky. But what we observed is cases who lived in census tracts that had higher levels of cadmium in the air had 2.2 times higher odds of residing in those hotspot neighborhoods. And we adjusted for all sorts of known risk factors, so it's not explained from things that we know about and can account for, like age, race, tobacco use, urbanicity, marital status, parity, insurance, family history, and other characteristics. And we also saw associations with ambient arsenic and ambient nickel. So this was published last year. We are currently working with the help of my postdoc, Dr. Johnny Newton, to look at, to tease out advanced stage diagnoses of breast cancer and hotspots there. So this is unpublished work, it's a work in progress, but I just want you all to appreciate that these maps are slightly different when we compare all breast cancer versus more advanced stages of breast cancer. So this is also with 3 additional years of data from the Kentucky Cancer Registry. Showing again neighborhoods in Louisville, but also in other parts of our state where I think we could benefit from some more targeted screening earlier on. So, this GETISORD, this fancy spatial statistic we can use this statistic to generate maps to hopefully promote better breast cancer screening programs on much smaller geographies than counties. And we can also use these to maybe generate hypotheses about potential risk factors. So I don't know everything about Kentucky, but if you know something about this place, maybe you can generate some novel ideas for why there are breast cancer clusters. So with future work with that LEAPS BRCA project, we're hoping to get more individual level data on residential radon exposure, as well as, instead of knowing what's in the air, wanting to know what's actually in the individual's urine, blood, or fingernail clippings. That allows us to measure things like those carcinogenic metals, as well as volatile organic compounds there are now, or that's actually been around for a while, these wearable devices where one can wear a silicone wristband to measure thousands of chemicals, such as dioxins, which I didn't get into that area here, but is also intriguing as potential risk factors for breast cancer subtypes. So overall, I think it's important that we study breast cancer prevention when we're thinking about metastatic disease. And there's still so much to learn about the role of environmental exposures as they relate to breast cancer subtypes specifically. I also want to highlight that utilizing GIS, or Geographic Information Systems, and registry data can help us to identify small area clusters of breast cancer, especially more advanced diagnoses, that I think we can use as a community to help us target screening programs. This has been a long time coming, with some major pauses. When the breast cancer work, a lot of this cancer hotspot work was done by myself and Tyler Ellis and Matt Ruther, and also students who have now gone on to graduate out of the Department of Epidemiology, Dr. Johnny Newton, Dr. Felicia Pugh, Dr. Maddie Tomlinson. We have great collaborations here in Kentucky with the Kentucky Cancer Registry, such as Dr. Jacqueline McDowell. I'm really excited to start our work on the LEAPS breast cancer team whenever that IRB approval gets through. This is obviously a huge endeavor and could not have been even fathomed without the participants in the Nurses Health Study and the Nurses Health Study 2 cohorts. So, again, their support outlives them. This research support was also funded with a pilot award by the CIEHS, or University of Louisville Center for Integrative Environmental Health Sciences, and will be funded with a joint School of Medicine, School of Public Health pilot project program. So with that, I really appreciate your all's time. Thanks for listening to me. I'm happy to take questions and answer what I can.
Such a cool presentation. So I have a question. So I've had a couple of cases where a patient has come to me and been like, this is really frustrating. My neighbor has brain cancer. My neighbor 2 houses down has this cancer. Like, what is the relation? And I think that, we do genetic testing, and a lot of times the answer that they perceive anyway is, this just happens, like it's chance. I have another situation where an individual has a workplace, and multiple individuals in the workplace were diagnosed with cancer, and so it's just a question that they have. Would you guide people to any sort of like resource or specific plan to say, hey, if this is something I'm noticing, what can I do about it? What can I test? What can I... Do you mind going to the mic?
Yeah.
So the virtual people. Yeah, sorry.
No, thank you. So I think sometimes the answer depends on the type of cancer, but I think also I think we need to do better at that in research and in public health in general, especially for things that we don't know what is actually causing. There's plenty of cancers where there are environmental, asbestos, radon, and lung cancer. Those are like great examples. And so I'm going to focus on breast cancer here, but I don't think it's necessarily considered to think about the environment and common cancers like breast or colorectal even. So I don't think we really have great resources for specific cancers, but if you're kind of thinking globally about any types of cancers that could be environmentally related, the EPA used to have really great information, like about radon, for instance. VOCs, I don't actually know if those are still what the state of those were, but I think the Environmental Protection Agency had great resources, and so does the CDC. It's a really hard question that I hope we can figure that out better, and I'm just maybe not the right person to give exact answers, but yeah.
So great presentation, very interesting. I have roughly 3 questions.
Okay, I'll get my water.
I'm assuming your hotspot analysis was based on incidence rates, right?
Correct.
And probably, as you know, so interestingly, breast cancer incidence rates are higher in non-Eastern Kentucky, but mortality rates are higher in Eastern Kentucky. So have you done an analysis using mortality rates?
Let's see, that was something we were doing with colorectal cancer. We are a small group, so I don't think we did breast cancer mortality, but that was part of the story that we're trying to get at with the advanced stage breast cancer. I think it's a little bit harder to do mortality hotspots because it might just be presenting differences in treatment and access, which is an important thing to describe. We were mainly focused from a prevention lens. So I think we have to just kind of be careful when we do mortality. Are we framing it correctly for breast cancer? And that story would be different for lung cancer where, so we have not done the short answer. We have not done the breast cancer mortality hotspots across the state yet.
Could be interesting to look at.
Yeah, definitely.
So I have other questions related to this map, so it's great to have it here. So I'm not entirely sure how you did your analysis. I've been involved in some of this kind of work.
Oh, great.
So your non-hotspot, what about, so that's neutral or non-significant. What about cold spots?
Okay, so we are not showing the cold spots. So, it sounds like you're familiar with Geddes or GI Star then. Okay, so it is what the statistic is, it is taking your census tract rate of, this is advanced stage breast cancer, comparing it to the census tracts that immediately neighbor you. So we used a Queen I can't say that word right now, around the census tract, and compares that to the global rate of Kentucky. So these areas are with 95. We could have also presented 90, depending on what the goal is of these maps. If we had all the resources, maybe we'd go down to something a little bit lower to more communities. We didn't go into the cold spots mainly because we weren't totally sure if they were truly cold spots, because a lot of these are rural census tracts and can have small sample sizes, and we couldn't tease out, is it truly cold or is it, so that was one of the things that
it could be low rates of detection.
Right.
So my other question was, what's going on in southeastern Kentucky there?
I don't know yet. I don't know.
So are you planning on, could you overlay this with mining areas?
We could. Our colorectal cancer maps are very much in line with a lot of mining spots, actually, in the urban centers of what we think of rural Kentucky. But we did not see it here specifically with breast cancer. But yes, that is possible if someone has like a GIS shape file, that is potential to overlap, yeah. And we can approximate distance. And I think with the coal mining things, what's nice about the EPA ambient data is we have data since 1999. So it might get pretty complex with the coal mines closing. You know, the timing of everything might be problematic, but certainly hypothesis generating.
Natalie, great work.
Thank
and I have a question about specifically about the radon exposure. Louisville is one of the hot spots in your study. I'm just giving a scenario. In a house where a child is growing and staying up to 16 or 18 years with parents, don't have resources for radon detection or mitigation. If the child is already exposed or the adult is already exposed,
Yep.
what is the antidote?
Well, I guess I'll speak from personal experience. My home that I grew up in, that my mother had breast cancer in, our radon level was 6 picocuries per liter, above the EPA threshold. But people down my street, their radon was below the EPA threshold. It is wildly variable. And also now I think it's a bit more common to have radon remediation. So what I actually think is probably one of the biggest, like, I know we don't have like a one cancer cure, but to think of how hard it is to get people to quit smoking and to think of all the resources we put in there and to think of a radon mitigation system roughly costs $1,000 to $2,000 and can be done in a day. And again, the state of Kentucky used to test all homes for free. I actually think this is not as financially and so long-term to fix. And also, you know, radon is a known lung cancer carcinogen. We could actually already be doing this from the lung cancer perspective, and I think that's another colleague wants assistance with that from lung cancer's perspective. But I think this is like actually, in my view, maybe I'm just a little bit too optimistic, but I think radon mitigation, if it's truly causing cancer, like breast cancer, could be a fantastic intervention that, you know, I can't necessarily modify my ability to breastfeed. I can't actually modify a lot of things that are those modifiable risk factors, but I can modify my radon with relative, yeah, I think there's a lot of interesting avenues that we can think about. Can it get covered by insurance plans? I don't know. That's for us to kind of decide as a culture and society, but yeah, you're welcome.
So every time we talk about data like this, I feel like I need to go measure my basement's radon and buy an air purifier on Amazon or something. It's still Prime Day, I think. Is there a way to quantify?
Today's the last day.
Yeah, maybe I'll do that for this. Is there a way to quantify longitudinal exposure? Because I know you can look at the hotspots per county or where they, you know, grow up, and it sounds like the nurses study, maybe they do follow kind of migration pretty well. But is there a way to quantify that? It just seems like it'd be really hard to do.
Sorry, I guess there's kind of two things about does radon stay consistent over time, and then another thing, people move. And so yes, the Nurses Health Study tracks people's movement. That's because they're mailing the questionnaire. So I didn't tell about that detail, but it was a cumulative average based off of where they were. So you know, if I'm in Jefferson County and then move to Lexington or Fayette County, it gets updated over that course of the follow-up. I should probably put those details back in here. But for radon over time, there have been some studies that have looked at radon over 2 years and over 10 years, and it's pretty stable over those long time windows. But like within a year, radon is really variable with like rain patterns, seasonal patterns, flooding. And there's also differences in like housing structures. So new homes, they don't have as porous foundations, so it can actually trap radon that does enter the home. So it's so variable, which is why it's really important that we need to like figure out the individual level. Like, yeah, we could live right next door and maybe have really different readings of radon in our home. Yeah.
And then for funding for these kinds of things, is it only through the government and like the NIH or whatever, or are there private sources for this kind of stuff?
So the reason I got into this work is when I went to school, my mentor, Rula Tumimi and Francine Layden, got a grant to study environmental exposures from air pollutant perspectives and breast cancer outcome within the Nurses' Health Studies. And that was through the Susan G. Komen Foundation in 2014 or 2013. But I have not seen a specific call from the government. I forget how many years ago, maybe like 5 years ago, there was more interest in like registry-based and like GIS and residential histories. Yeah, but I don't know how it'll work.
The reason I'm asking is,
either,
we talked about the pucks for radon detection and like the bracelet
Yeah.
clippings and all that. It doesn't seem like it's that much compared to $150 million for a clinical trial.
Right.
And there's a bunch of people behind me that tell me they have money for breast cancer stuff.
Oh,
I
okay.
don't know if it's gonna work out, but-
Well, and I do think it's something like a little bit hard to convince reviewers. Like, I sat on an NIH grant review panel, and it is very wet lab heavy, like petri dish, a controlled experiment, and rightfully so. That's like the scientific process. But unfortunately, humans are not inside petri dishes and have a lot of variability. And I think it's really hard to convince reviewers to fund these enormous. I know our study is small, but like if we wanted to do this in the Nurses' Health Study, it would recreate a cohort. It's millions. It's so big, and so I think if we can just like move at least incrementally, maybe that's when the reviewers will be like, oh, this is actually something. But or maybe I just need to write better. I don't know.
Trying to figure something out.
Yeah. Cool.
I do have one question.
Yay.
Do you use any type of artificial intelligence in your
No. In this
Study.
work?
in your work? Yeah.
No.
Think that would help, like with the data?
Well, you were definitely not allowed to like upload data to AI. But maybe with like summarizing, actually for a grant, I had like 5 pages written about radon. And I was like, can you do this in 2 paragraphs? And it did a great job. I had to like edit it, obviously, but
Right.
I'm kind of, I'll admit it, I'm like a AI skeptic. I feel like my brain I need to keep it working.
Yeah.
But I don't know. If I knew how to use it better and like adequately, I'd like somebody teach me. And yeah,
I think you more like data analysis, though, right? Is there, I don't know, I'm not an epidemiologist, but like in terms of overlapping these maps and that kind of stuff.
yeah.
Is anyone using that for any setting yet?
In AI spaces, there's definitely like an AI for GIS. I just don't know anything about it. Yeah, good. Yeah, I don't know. It's actually GIS, if you know what you're doing, it's a very good interface. So like, doing the actual stuff in GIS is like it's getting the data into GIS in the way that's appropriate. That's the harder part, but I guess what it could help me with is just like batch overlaying probably in ways that, and probably has like all these access to maps and things as long as I know if the maps are good or tested. Yeah, it'd be really cool.
So I have a very long, no, it's not one. So I have a background in epidemiology as well, and I always
nice.
wonder about studies like the radon studies because, as you point out, they're very complex, right? To really know how truly people have 300 millirems of exposure of radiation just if they're a gardener. So there's so many things that happen if they fly in a plane, all those things. And I'm just wondering if some of these things would be better if we looked at, and we'll be the speaker on genomics and genetic mutations and familial history and stuff, if we try to overlap maps to better kind of start leading you to see who has just, in some of these areas that they probably, and forgive me, I'm not from Kentucky, so if I say anything that offends anyone, but I'm assuming that many of those people lived there for multi-generations and that there's probably a concentration of genetics. And it would just be interesting to see some of this, because the radon, I'm gonna go out on a limb and say radiation exposure is bad, so we can just make that assumption, and just what are those other things that help drive that, and how do you quantify that? And secondary smoke, it's all those different things. It would just be interesting.
Yeah, it really would be. And yeah, and I think these maps can like be really helpful to like think about those things. And I've tried to like steer it more about like screening, because you're right, these maps don't tell me about a genetic cluster or, but if there is high breast cancer, especially advanced stage, like we can feasibly go out there and screen. But yeah, we are actually working from a colorectal cancer perspective, working, there's, I guess there's some funding from Foundation that have done like genomic screening on the subset of Kentucky Cancer Registry participants. So, and there's actually breast cancer data out there. I just don't know how many, but we're starting to dabble in like getting the IRB approval, because right, the mechanisms of how this all work are, you know, I showed 2 Beagle studies and like 3 cell lines. It's so minimal compared to, you know, what we know. And yeah.
Instead of looking at just people impacted, they actually looked at all like benign and pre-malignant
That's
tumors
great.
and saw microsatellite instability in a wider array of people than they imagined. So that kind of thing in the breast cancer realm may be interesting as well.
Yeah, definitely. Thank you. Yeah.
I'm assuming you have demographic information on these people.
Yes. Yeah.
Did you show that?
Well, these are age standardized. So what you're seeing here is not due to age, and they're all females, but we couldn't really we couldn't standardize beyond that without parsing the data way too thin. So this is, the map you're seeing is age standardized. So what you're seeing is not due to one census tract being older, but yeah, we do have demographic data. I'm just showing a snippet. Our paper has like all of them. I'm just picking out ones because it was a ton of numbers, but yeah.
So did you check for significance? So particularly, a lot, if you go back to the map, a lot would be Caucasian, but particularly in Lexington and Jefferson County, what about race and poverty?
And that's where we were describing them. We couldn't like account for those on the map level. What we could do was just describe who lives in those places. And that's what, there were, of the, you know, roughly 77,000 females with breast cancer, 5,044 lived in one of those breast cancer hotspots. And among those 5,000, 17% of them were Black, whereas the people who resided in those non-hotspot areas, only 6% of them were Black. We also, you know, see differences in marital status too, as well as parity. So the people residing in the breast cancer hotspot, 14% of them were nulliparous, whereas people not residing in those hotspots, 9% were nulliparous. So there's actually, it could be more than demographics. It could be what we already know as risk factors. Yeah, so that was what we attempted, but we couldn't really do it on a spatial level, unfortunately.